Today we will discuss about how the immune response work against the non-self epitope. So when the immune system within a host detect a non-self epitope, how it will respond. So one thing before we proceed into this topic, I would like to say is that we will not discuss in terms of T cells and B cells, and not in terms of how or what are the first defense and how this secondary defense or more specific response against the non-self epitope generated. That topic we have covered many times in our previous videos. You can refer to those videos for that understanding. In this video we will cover a general overview how these immune cells reach, how these immune cells actually responds once it detects a non-self epitope. So these are been uh, this black color shaped structures are being considered here as the danger signals, pathogen or non-self epitope. So once they enter the host cells, the there is an immune response, that is homeostasis, or that is hematopoiesis, and tolerance will get disrupted, and it will trigger the mobilization of the immune cells to the site of these pathogens that can be seen here with the help of blood vessels. And once these immune cells reach near the site of these infections, the extravasations of the immune cells will occurs, and because of these chemokine gradients, there is a migrations to the site of inflammations uh, that leads to the immune cells reach at the site of these inflammations or where these non-self epitopes or danger signals are present. And once they reach to the site of inflammations, they will interact uh, with this danger pathogens, or the interaction between resident cells, immune cells and the pathogens occurs here. And after that, the immune cells become activated and release the cytokines that will trigger apoptosis. That can be seen here. The effector functions on inflammatory cells, the apoptotic effect of the epithelial cells that are infected with these danger signals are being triggered, and also this apoptosis will be triggered against the cells, against the danger pathogens that are already inside the host cells, or that has already passed through this epithelial layer. Simultaneously, because now the immune system has the information about this non-self epitope, so the more specific immune response will be generated in terms of the antibodies that will work against those pathogens. And so these antibodies will also start to act against those pathogens, that is effector functions of inflammatory cells that can be seen here. So here we can say that more generic response of triggering the epithelial cells that contains the, the pathogen signals, and here we can think of it as a specific response in terms of the antibodies that can easily distinguish this non-self epitope, or antibodies that are generated against this non-self epitope. So after that the target cell, after it will effector functions will trigger the trigger or effective functions will act against those danger pathogens. Then the cells that are affected with those pathogens will will be triggered, or cells those are affected with these danger pathogens will undergo apoptosis because of the inflammatory actions or effector functions by the immune cells. So, and one of those triggering of apoptosis might be occur either by the release of cytokines that we have discussed in our previous videos, and also sometimes to the intracellular or to the cell-cell interactions that will trigger the extrinsic apoptosis. So I hope you got a basic idea what are the how the immune response works whenever it immune system of the host detects non-self epitope. And each of these topics descriptions, each of these step descriptions are presented in this table on the left hand side that I have just covered in a normal normal terms. So these are more of like a definitions of each of the terms, and how what does this actually means that we have already discussed. And if you need to understand more about this immune response against the danger signals, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.